Hi, everyone. I'm Joe Tucker. I'm the CEO of Enveric. We are working in the psychedelics, or I'll say the non-hallucinogenic psychedelic space, which we're now calling neuroplastogens. The idea being to take on, as we just heard, and I think as we all know, in this space, there's a huge unmet need in mental health, both depression, anxiety, PTSD, across the board. And a lot of the reason that we saw in the last few years, this great deal of excitement around the psychedelic space is because of the very large responses that we were getting in these initially academic and now corporate-run trials. But one of the questions, of course, is what's really going on, what's underlying it, and it's when you get the hard scientists started to ask those questions in the last few years that this new concept of induction of neuroplasticity and the molecules behind that being now known as neuroplastogens. This has only come out in the last few years. So if you don't know it, briefly, the concept is in the brain, we have different regions responsible for different things. I'm not gonna get too detailed for the purposes of this conversation. But imagine, for example, the amygdala, an ancient region of the brain responsible for taking stimuli and turning into a fight or flight response, for example. And then you have other regions of the brain, like the prefrontal cortex, much newer, whose role really, again, to really simplify it, is to be cognitive about it and say, should I be running away from this stimuli? And the connection between these two, the balance of the connection between these two is what you have in a normal brain or a healthy brain. And what we started to see, again, when people were trying to understand what's going on in the psychedelics, what's going on is the patients that have atrophied, let's say, connections between these regions of the brain are finding they're getting this repaired. The neuroplasticity, the induction of the neuroplasticity does not create new cells, does not create new neurons, but what it does is it enhances the connection between these regions of the brain by stimulating increased connectivity between the neurons. So we got pretty excited about this. We looked into this class, and I'll admit, we originally, at Enveric, we had a hallucinogenic agent, novel compound in this space earlier on, but we saw some of the problems coming that I think everybody saw, anyone paying attention, with Lykos, MDMA. We have the functional unblinding issue. We have a lot of commercial challenges. How are you going to scale this? It's very expensive with the requirements for infrastructure, the requirements for oversight, clinical oversight. And at the end of the day, the regulatory question is a pretty big one. Are we going to actually get there? One thing I can tell you is all the conversations that we've had with Big Pharma over the last few years, very little interest on the part of Big Pharma on a psychedelic agent, very little. But if you can solve that problem by taking the hallucination out of it, suddenly there's a lot of interest. So we said, let's go after what can we do? We started with the original, these receptors, the 5-HT2A. Well, 5-HT is serotonin. So you start with serotonin, you make novel molecules based around that core structure and say, can we get it to bind to, and these are not the novel molecules, this is illustrative purposes only, but we made really more than 1,000 different novel molecules and said, it's a pretty big leap, can we segregate the hallucination from the induction of neuroplasticity? And we found that it's not a common thing. Most of the vast majority of the molecules we made, if they hit 2A as an agonist, they cause neuroplasticity, but they also cause hallucinations, the majority of them. We did find a few that cause hallucination, no neuroplasticity, interestingly. And the reverse, of course, is what we're looking for. Ultimately, we came up with a molecule we're calling 003, was not the third molecule we made. It was more like the 900th molecule that we made, but that hits our TPP, that hits our immediate efficacy, which is partly why people are excited about psychedelics, but has the low side effects, neither what we're now considering a side effect, the hallucination, the experience, is neither that nor all the other challenges we're seeing that we all know about with antidepressants, the existing ones. Now you don't have to have the treatment in clinic. This can be much more like a standard pharma-friendly drug. And this, of course, gives you access to a much greater patient population. Ultimately, we still want to see that induction of neuroplasticity just as you do for the psychedelics, and that's what we see with our molecule. So we're early. We are preclinical right now. There's actually only, I believe, one neuroplastogen at all that's gone through a phase 1A trial at this point. It's Delix's compound. Of course, we're all very excited watching what's going on with Delix. But we are still in preclinical here with our molecule. Should be some interesting stuff happening to us really more in 2026 and 2027, where we answer the two big questions. Question one, and these are the questions we get from all the big pharmas that talk to us: is your molecule really non-hallucinogenic? Sure, it doesn't cause head twitch in rats, but what happens in humans? And then the second, without the hallucination, does it actually work? And if any of you were on my panel earlier today, that's the big question we all don't know. We all think probably, but we don't know until we do it. So that's what we're working on right now. Very large market for us. Of course, we know it's a huge antidepressant, anxiety. These are very large markets right now, only expected to grow larger. In particular, with neuroplastogens, we see a potential not just in those markets, as monotherapies, but potentially as enhancing the activity of the other molecules already in the space. Now, in our path, Enveric's been a public company for just about 4 years now. Accidentally, the company working in the psychedelics was acquired, and we suddenly found ourselves this public company. As we went through this last few years of the quest for a non-hallucinogenic, and it took a while, right? It was not easy, and there's no model out there. Nobody knows what the structure should be at all. So in the last few years, we created quite a few molecules along the way that have other interesting profiles. And we've managed to out-license a few of those. As you can see, as a small company, we're entirely focused. We've not received to date sort of the $200 million funding, sadly. And so we've had to focus entirely on the molecule that we think has the greatest possible impact for us. So that's 003, the non-hallucinogenic psychedelic, and some of the other molecules we've out-licensed along the way. Management team, I won't bother going into it other than to say we do have on the board a number of folks from Big Pharma, which is really helpful to help guide us. We are really paying attention to where the receptor industry is for us, and all those conversations with Big Pharmas have convinced us that non-hallucinogenic as a psychedelic, as a neuroplastogen fits, but psychedelics do not. There's no interest that we've seen there. So I'm going to leave here because I know the last two ran over. So I wanted to stop a little bit early and give the audience a chance for any questions if you have any, or you can get back on track. Thank you.